In the article, DNA May Offer Rapid Road to Zika Vaccine by Megan Rosen, published February 28, 2017 on ScienceNews.org, the process of constructing, using, and the future of DNA vaccines for the fight against Zika is delved into. DNA vaccines offer a broad scope of advantages, including the speed at which it can be created, the safety of the procedure, the flexibility in its composition for an enhanced immune response, and the newfound success in human trials. Inserted into a plasmid is the Zika genes, which then journeys into the nucleus of a cell and aids in the process of making messenger RNA. This messenger RNA will trigger the immune system to produce antibodies to aid in the construction of Zika proteins. DNA, or deoxyribonucleic acid, is comprised of molecules called nucleotides. Each nucleotide contains a phosphate group, a sugar group, and a nitrogen base. The four types of nitrogen bases are adenine, thymine, guanine, and cytosine. The order of these bases is what determines DNA's instructions or genetic code. It is made up in the form of a double helix with strands headed in an anti-parallel 5' to 3' direction and is held together by hydrogen bonds. To construct a DNA vaccine, you must first synthesize genes from a pathogen, which is a bacterium, virus, or microorganism that can cause a disease. Then, you have to inject the synthesized genes into a circular, double-stranded DNA molecule in a cell that can replicate independently of chromosomes called a plasmid. Plasmids that are used in labs are designed to introduce foreign DNA into another cell. After making many copies of the DNA, the vaccine is ready for trial. For the DNA vaccine to work, it has to trigger the immune system to make antibodies to respond to the virus. To do this, weakened but not dead Zika genes are inserted into the DNA plasmid. A gene is a sequence of nucleotides which forms part of a chromosome. The nucleotide order determines the order of monomers in a polypeptide or nucleic acid molecule which a cell or virus may synthesize. The Zika DNA plasmid travels to the nucleus. A nucleus is a membrane-bound organelle in eukaryotic cells that contain the majority of the genetic material of the organism. A cell's replicated DNA is transferred from the nucleus to the mRNA, which then codes for proteins. mRNA is a subunit of RNA that sends genetic information from DNA to the ribosome, where they specify the amino acid sequence of the protein products of gene expression. In this case, mRNA produces Zika proteins that form virus-like particles. These particles then activate the immune system to make antibodies to make Zika. Antibodies combine chemically with substances that the body recognizes as foreign, such as bacteria, viruses, and other substances in the blood. Number 1. What are the four pairs of DNA bases that form the double helix? Adenine, guanine, cytosine, and thymine. Number 2. What is mRNA? mRNA delivers the genetic instructions of DNA to ribosomes.